name is Jane Farrer, and I'd like to take the opportunity today to talk about the Human Genetics degree course in the School of Genetics and Microbiology in Trinity College Dublin, which is part of TRO 060. So during this short presentation, I'd like to talk about what is human genetics, describe a little bit of what is human genetics, and to talk a little bit about what is studied during the Human Genetics degree course in Trinity College. And after that, to elaborate a little bit on some of the exciting research that's ongoing in Trinity College and globally around the world in human genetics. So firstly, what is human genetics? So each of us is composed of about 40 trillion cells. So in our human bodies, there are about 40 trillion cells. And inside every cell, the nucleus of the cell has the DNA or the human genome in there, the code that is determining the function of that cell. It's giving the instructions to allow the cell to make the right proteins so that that cell works really well and functions really effectively. So that's the human genome code inside every cell of our bodies. So each of the 40 trillion cells has got that code in there. So as I said, that's called the human genome, and it's comprising 3 billion letters of code. And that 3 billion letters of code is found on 23 pairs of chromosomes. So those 23 pairs of chromosomes that we have in every single cell of our body has got the 3 billion letters of code. And in fact, if we took the DNA in those chromosomes and from each cell and spread it out, each cell would have 2 meters of DNA. Of course, we can't do that, spread it all out in a line, but it's about 2 meters of DNA in every cell. So 2 meters of DNA in 40 trillion cells. So we have got absolutely hundreds of thousands of kilometers of DNA in our bodies, which is a phenomenal thought. We are really sophisticated pieces of machinery, biological machinery. And sometimes we call the 23 pairs of chromosomes the human karyotype. So the human genome is the 3 billion letters of code on those 23 pairs of chromosomes, and the 23 pairs of chromosomes are termed the human karyotype. We have made unprecedented progress in human genetics over the last almost 2 decades. If we look back and see that the first human genome was sequenced in 2003, it took many, many years, many institutes all around the world, thousands of people working on it. But today you can get your genome sequenced for about $500, and it doesn't take very long at all. So there has been an amazing progress in understanding the human genome and in getting genome sequenced from thousands and thousands of people. So now we're understanding the very small differences that occur between humans. In fact, about 1 in 1,000 letters of the DNA code differ between you and me, between you and an unrelated individual. And some of those differences are silent and have no effect. But of course, some of those differences predispose us to disease, increase our risk of disease, or sometimes they actually in themselves, a single change in a code, a single mutation can be causative or cause a disease. And we're beginning to track on those. And the power of that is not just for diagnosis of genetic disease, but it's also for designing novel interventions or novel therapies. So as I said, there has been unprecedented progress from 2003 when it cost about $3 billion to sequence one genome. And that's because there was so much work and the technology had to change and upskill so much, to now in 2020 where it's $500 to sequence a genome, and you can get your whole genome sequence back in a matter usually of a couple of weeks. So there is unprecedented progress, and we're really in the era now of generating big data with lots of human genome sequences from lots of populations, comparing, for example, normal, healthy groupings to the genome sequence of people with diseases to try and track on what are those genetic changes that really predispose us to disease. And so for that reason, we've really entered the era of medical genetics and personal genomics. And so I'm showing this slide here of Angelina Jolie, who's a well-known actress, because a number of years ago, she very bravely told the world that she actually carried a breast cancer disease mutation. And so she was at significant risk of breast cancer. And really, she told everybody that you can be empowered by genetic information, and you can decide to do something, to intervene, if you understand what potentially what disease genes you may have inherited, and therefore may increase your risk of certain disorders. So that's the whole area of personal genomics and the power of genetic information. So the second thing I wanted to talk about was what is studied during the Human Genetics degree course in Trinity College. And actually, the course is 4 years in length. The last 2 years, you will specialize in human genetics. So in third and fourth year, and we'll spend your time in the building here on the right-hand side, which is the Smurfit Institute of Genetics. The genetics department was founded in 1958, and its philosophy is still, to this day, a research-led department. So most of the principal investigators and lecturers in the department are involved in cutting-edge research themselves. And so you will be taught by people involved in the area of human genetics in cutting-edge research projects. So the course structure in the first and second years are the junior freshman and senior freshman years. There you will cover many areas of biology, including genetics, but many areas of biology. Chemistry, some math, statistics, computation, and also we'll have choices of approved modules. And that's really for year 1 and 2. I'm just showing year 1 here, the course structure, but similar for year 1 and 2. In the third year, our junior sophomore year, you will specialize, as I said, very much more in human genetics. So third and fourth year, you're specializing in human genetics. And you will get a foundation in many areas of human genetics and many areas of genetic analysis. In addition to that, you will be able to choose approved modules and also Trinity electives, which are in completely different disciplines. So you'll also have quite a lot of choice. During the junior sophomore year, then there is an opportunity for a field trip with faculty, and also there is an opportunity to compete for summer internships. So in human genetics, there's an opportunity to compete for summer internships which will happen in the summer after third year, and they will happen in international labs. The final year course structure involves, again, more advanced modules in human genetics, medical genetics, and genetic analysis, and also a literature review of a topic normally of your choice, and a research project, a 10-week capstone research project, where you will become part of a research team, for example, a team in genetics, in human genetics, and then you will undertake a lab-based or computational-based research project, and then write a thesis. And what do our graduates do? Our graduates are really sought after because they've learned a huge array of skills during the process, to problem solving and to hard work as well, an array of skills during the degree course. And what we find is more than half of our students go on to do a master's or a PhD in a related area to human genetics, so in human genetics, because in fact it's a really addictive subject. Once you get into it, and because so much is happening now in this era, that it's really something that reels you in, and our students tend to want to stay in the area. And then they often become academics, and we have professors that were in our department all over the world, contributing to the body of knowledge in human genetics. Other students get involved in the pharmaceutical and biotechnology industries, which all use genetic methodologies and techniques, and also get involved in campus companies, the generation of campus companies. For example, a number of campus companies have emerged from genetics, including Identigen and Genable technologies. Others go and do postgraduate medicine or law, are scientific journalism, teaching, work in government agencies, and many other careers. But importantly, our graduates are really sought after because the really significant skill base that they have developed during the degree. So the last topic that I wanted to cover was, what are the powerful and exciting applications of human genetic information? We could go on for hours about this, but let's focus on a few areas that are happening within the genetics department in Trinity College, but also globally around the world. We're going to look at a couple of examples globally too. So within the genetics department, there are teams interested in actually ancestry, because we can look and take material from very old bones and look and interrogate populations, for example, the Irish population, using the material from those old bones. Usually the genomes from those old bones, the DNA is not good quality, but you can stitch together genome sequence from them and understand more about Irish populations, for example. So for example, Newgrange, some of the bones that have been found in those Neolithic tombs have been used to understand more about the populations in that era. And it seems there may have been a ruling elite class because one of the bones that was taken from a male in Newgrange, it was obvious that that was a child from clearly an incestuous relationship. A close relationship, which would suggest, an elite ruling class. And other teams in the Department of Genetics are interested in programs of cell death and also in the programs that drive tumorigenesis or cancer. And in fact, our cells are programmed obviously to live, but they are also programmed to die appropriately. We don't want cells exploding in our bodies. When a cell has lost functionality, we want it to die kind of gently without causing inflammation. And that program is called apoptosis or programmed cell death. It's also used in development, in utero a lot. But some cells have learned to avoid that program of cell death. And so cancer cells have very cleverly learned to avoid programmed cell death. But the programs of cell death and avoidance of cell death, have genetics behind them. So there are genes and encoded proteins involved in all of these processes. And clearly, if we understand a lot more about those genes and what they're doing, the encoded proteins, then we can think about novel and innovative ways to design therapies, tackling, for example, things like cancer. And there are people in the department very interested in that. There are lots of other groups as well in the department that are actually tracking down disease genes. So really investigators, but genetic investigators, trying to understand, what are the genes and encoded proteins that are actually causing disease in our populations. You can compare a population of normal people to a population of people with a disease and see what the genetic changes are. And because we now can sequence whole genomes very rapidly and very effectively, we can take large populations of normal people and large populations of people with a disease, for example, motor neuron disease, and ask what are the genetic changes that increase your risk of getting one of those diseases. And then sometimes we also work, for example, on disorders that actually are due to single genes. And in this case, I'm talking about ocular disorders. There's groups within our department working on inherited eye diseases where people lose vision and they sometimes can actually lose all perception of light. For example, the panel on the right that is black, their world becomes black because they lose their photoreceptors at the back of their eye, their retina. And that's quite often due to mistakes in just one single gene. At a time, there are 300 different genes because there are many different inherited retinal degeneration disorders. But any one patient has a mistake just in one single gene, an encoded protein that's resulting in them losing vision. And we're tracking down those disease genes. But I think the power of doing that is obviously for diagnosis, but also for designing innovative ways to correct those diseases, to deliver back, for example, the normal gene, that's gene therapy, or to go in and correct at the DNA level, the mistake. That's called gene editing technologies and new technologies that have emerged. And there are other teams within the department that are interested in neurogenetics, so interested in brain function, in how cells, neuronal cells in the brain communicate together, how messages are communicated down axons and dendrites, and how the genetics underlying all those processes and making those processes in the brain. There are other teams that are interested in what we call the blood-brain barrier. So it's very important that things cannot enter from the blood into our brains, because we don't want to get inflammation in the brain. So maintaining an intact barrier, blood-brain barrier, is very, very important. And again, there are genes and encoded proteins that are very involved in doing that. A whole group of teams in the genetics department are interested in the blood-brain barrier and disorders that might be associated with breakdown of that blood-brain barrier. So finally, I just want to give you two examples of how genetic information is driving huge change in the whole area of medicine, in medical genetics, clinical genetics, and in the way we attack and understand disease and actually design innovative therapies. So let's think about haemophilia. Haemophilia is an example of a disease that was, always in the leaving, and is always in the Leaving Cert book. It was deemed as a royal family disorder because this one ran in one of the European royal families around the world. But in fact, a lot has happened subsequent to that in the whole field of haemophilia. So let's look at it. It's a really good example of how genetic information is helping us to change disorders. Firstly, we were able to track down that the genes that cause haemophilia were on the X chromosome or sex chromosome, and that's why males are affected with this disease. And when you have a mutation in the factor 8 or factor 9 genes, then you don't clot properly and you're at risk of life-threatening bleeds, or indeed, for example, bleed into a joint, as you can see in this knee joint here where there's a massive bleed into the joint, and that's a haemophiliac patient who isn't clotting properly and is getting large bleeds. So genetics helped us understand, those are the two genes involved. And after that, we used those two genes in cell factories to actually make the recombinant proteins, so recombinant factor 8 and factor 9, to give to patients so that they would be able to clot better because they didn't have the normal gene themselves encoding the normal protein. And so that's what most haemophilia patients are on around the world, on these recombinant proteins that are made using genetic technologies. But then recently, in the last number of years, people are saying, well, can we design gene therapies where we can actually deliver back to the patient the normal gene, and then their own body will actually make the proper protein, the factor 8 or factor 9 protein. And so small viruses, non-harmful viruses, are being designed with the factor 8 gene or the factor 9 gene. And they've been given to patients, injected through their bloodstream. And actually, we're now at the end of phase 3 clinical trials, and the data looks really good, where patients with haemophilia with once-off injections of therapeutic virus are making doses or amounts of factor 8 and factor 9 that is changing their disease, that they are now clotting appropriately. Very exciting time for haemophilia. And it's also a second example I'm going to give globally is the first ocular gene therapy, in vivo ocular gene therapy that was approved around the world, is a gene therapy for an eye disease. And this again is using a virus, a non-harmful virus, where the normal gene has been incorporated into this virus. The virus then gets injected into the eye just once, subretinally, under the retina, where your photoreceptor cells are in the eye. And the virus infects those cells and starts making the protein that is missing in a patient that has mistakes in the gene that causes this particular eye disease. And so we're actually going to look at at this gene therapy in action. We're going to look at a video of one of the first patients in the world treated with this gene therapy. It's a virus encoding a gene, and this patient is missing a normal copy of the gene because from their mom and their dad, they received copies with a mutation in them. So now we're delivering the normal gene to the eye by subretinally injecting it into the back of the eye just once. And we're going to see the response of that patient to that subretinal injection. This was developed in the University of Pennsylvania and also the hospital, the Children's Hospital of Philadelphia, by Jean Bennett, Al McGuire, Kathy High, and then the technology was further developed in Spark Therapeutics, and it was approved by the FDA and EMA in 2017 and 2018. So this can now be administered to human patients. It's been through clinical trial, it's got approval, and recently the company Spark Therapeutics was bought by one of the largest pharmaceutical companies in the world, Roche. And this therapy is now available for patients. So it's been through the clinical trial process, it's approved and available for patients. But what we're going to look at here is we're going to look at one of the first patients in the world treated with this therapy. And this is a little boy, and you will notice that he has one eye patched and one eye that is not patched. And the eye that is patched has been treated with an ocular gene therapy, a non-harmful virus engineered to have the gene that he is missing, a normal copy of the gene, whereas he inherited a mutant copy from mom, a mutant copy from dad, and he himself has no normal copy of the gene, and so he isn't making the encoded normal protein. This protein is involved normally in visual transduction. It's an enzyme involved in visual transduction of light. And so it's essential for vision. And gradually his photoreceptor cells are dying. Those cells at the back of his eye are dying. His vision is getting worse. He's currently working off his best vision because the eye that is treated and is patched was the eye that had the worst vision. Always in these studies, you treat the worst eye, the worst vision eye, because you don't want to risk any vision that he has. So this is the absolute best vision he had before his therapy. And he's trying to find his way and negotiate his way through a maze. Through a maze, he's trying to negotiate the obstacles and see his way through the maze. That's Jean Bennett, who was involved in developing the therapy, the lady in the picture, in the video there. And so he's trying to negotiate his way. And let me tell you what's actually happened in his other eye, even before we see the video of it. In this eye that is patched, he's had under the retina about 10 to the 11 copies of a virus carrying the therapeutic gene. He's had that injected subretinally into his eye. And that therapeutic gene has infected the cells in the back of the eye, and the virus has infected the cells, and the gene now is being used to encode and make the correct protein, so that he can hopefully see a lot better from the treated eye. But in the meantime, what we're looking at here is his untreated eye. We're understanding really how serious and significant his vision loss is due to this genetic condition. Simply a mutation in one gene encoding one protein, but involved in light transduction. And unfortunately, it has a radical effect, and his vision is highly, highly compromised and getting worse over time. And as you can see, he's trying to find the handle of the door. He can't see it because his vision is so bad. He's finding it really hard. He's struggling to see the door handle. And eventually he's going to get it now in a second. And now in this next video, we're going to look at this chap. And in this second video, he's going to be working off the treated eye, and his untreated eye is patched. So this is the power of having genetic information. This is one injection of a virus with a normal therapeutic gene into the back of the eye. It's now making the right protein that is working in visual transduction and allowing him to see a lot better. He doesn't have perfect vision, but he can see an awful lot better. after a single injection of the virus. Powerfully, years later, after that one injection, these patients that are treated have retained their improvement in vision. So really, I just wanted to demonstrate to you with this the power of genetic information, what we can do if we understand a problem, a cause of disease, and we can sequence human genomes now to understand causes of disease, then we can design novel therapies, whether it's providing a replacement gene, such as this ocular gene therapy, or whether it's actually correcting the disease using very recent gene editing techniques. So what I hope we've done in this little presentation is tell you a bit about human genetics, what it is, tell you what is studied during the human genetics degree course in Trinity College, and importantly as well, outline the really exciting areas of research in human genetics, both in Trinity and globally around the world. This is such an exciting era for human genetics. And with that, I'd just like to thank you for your attention.